Okay, so today our agenda is to go over some of the two-photon analysis that Davies and others have done to scaffold a figure. Um, Last time we discussed this, we agreed to sort of I laid out a potential structure, and so the intent, yes, this is this figure. So we are in the process of making the data release paper, and most of the method is written, and so now we are focusing on a subset of figures that highlight the data and then some first order analysis. And so this figure is intended to introduce the units that are extracted from all of the modalities. And so today we're focusing on the mesoscope column. And so I was hoping that Davis could help us have this discussion today. And you said you had some material to share.
Yes. Um, yeah, so uh, I'll go ahead and share my screen. Um, okay. Yeah, so I think our from sort of picking off from our last meeting, we had talked about some different signal to noise measures and sort of trying to split those up into different things to kind of get better clusters of um, uh, you know, good traces. And we kind of found that using, while I was going through those, that um, many of um, uh, the metrics that we found, we ended, they kind of yielded very similar um, issues. And they also had, we ended up kind of evaluating them based off of their the event matching, how well they kind of correlated with these sort of measures of how well the events matched these certain exponential and Gaussian noise expectations, and that we were ultimately kind of just using these visual things to evaluate them anyways in terms of just looking at the traces. After a suggestion from Jerome to actually kind of get a background measure of an individual trace and then use the standard deviation of those events from that background trace to kind of see for a given trace, are there, um, what proportion of their events that are detected using Allen's sort of OASIS style deconvolution method, um, what proportion of those events are, four standard deviations, 2 to 4 standard deviations, or two. So I kind of use that as my starting point. And the way I sort of defined the kind of background measure I'll get into here. So basically, we took kind of a window around all of the events. So zero point 0.5 seconds before and 2 seconds after, which is maybe a bit of a long um, window, but and it yields pretty good results, but there may be a reason to shrink it. I haven't done that yet, but I can get into that later. And so defining those as kind of like event windows, and then anything that wasn't in an event window, we calculate as the background, and then use the median absolute deviation of that as like our noise um, or our sort of like background measure. And so then once we calculate that background measure, we then use that as the basis for our for a given event. How is it above that measure? Um, and so got some of the plots down here for that.
Can you give me a little more? I'm not sure I completely understood.
Yeah,
How
sure.
is the event defined? When you said
So
window,
yeah, so the events are if there's it's 2 seconds after the event window is like so the point so if the event that comes with the that's identified by Allen and Oasis style
oh, I see.
deconvolution, 2 seconds after that event and 0.5 seconds before it. And if events occur within zero point 0.5 seconds of each other, which happened a lot in the data, then we would merge them into one event and take the midpoint of all of them, which led to some uh, it mostly works, and then there's some issues where you have like, a worst case where there's like a few that are all right at the border, and then you have like an 8 second um, event window, which doesn't make sense, but there was only one of those that occurred um, out of all of the ones. So I think that's something we can definitely adjust. Um, does that make sense in terms of or does that seem like a good approach in terms of the
Yeah,
like,
no, that makes sense. I think you try
yeah,
to find the background outside of the event if it makes sense. It's
okay.
also pretty intuitive, I think. But yeah.
Okay, gotcha. Um, and so we get some uh, plots for these in terms of the traces that we have. Well, yeah, this sort of goes over sort of what I was just talking about. Um, so we get
Can I
a,
ask, David? I'm very sorry. I was 8 minutes late. I was in another meeting. But the event here, you mean like kind of like the inferred spikes, right?
yeah, it's the inferred spikes from Allen. Yeah.
So because you had written Oasis slash Allen. So now we're talking about inferred spikes, not using Oasis, but using the Allen method.
Yeah, I think they basically use the same kind of underlying algorithmic approach, but just with different, I think, regularization methods. But it's like very similar from what I understand.
Okay.
Yeah, that's right. It's I think it's okay not to. Yeah, but that's fine. Yeah.
Mhm. Okay, so yeah, so we have this is kind of gives an example of the so like this would be sorry, the zoom is a little tricky. Um, of like two events that occurred within zero point 0.5 seconds of each other, or actually this is three, um, and then was merged to like a midpoint here. So with sort of more normal events like this, you don't, with there's not clustered together, there's no merging, but then with some like these, you get a couple more. So it kind of takes the average of them, the ones that occur very close together. Um,
I have a question.
yeah.
Um, What do you mean by averaging events that occur close together?
So it's
What do you,
basically because we're trying to define these windows of what is like an event versus what is background. And a lot of times, and so we're kind of trying to pick reasonable windows around the event that would reflect kind of like calcium decay for this indicator, and these types. And so you're going to have 2 seconds on. So we kind of, as a rule of thumb, do half a second before, because the rise is faster, and then 2 seconds after, as like of wherever there's an Allen detected event in the data. And just because of the nature of the algorithm, you end up getting a lot of events detected like right next to each other rather than just like one event that has like a higher amplitude or something. Um, and so the goal with this was basically to better be able to, so you don't end up with like huge swaths of the thing being counted as event space, like merging them into one when multiple events occur very closely together, merge them into like one window, basically, if that makes sense.
And just to clarify,
Yeah,
you are defining an event as a, I know that was mentioned as a, how are you defining an event? Can you
it's anything that was
reiterate?
detected by this sort of Allen Oasis style deconvolution algorithm. And
Which
those, yeah.
corresponds to spikes, presumably.
Yeah, so it's kind of seeing because what we noticed is that our kind of heuristic for how we were evaluating our other signal to noise metrics was basically seeing whether they correlated with something that visually looked like a calcium transient that was clear versus something that looked like a lot of just random fluctuations. So we kind of decided we might as well cut out. I guess the approach here was like cut out the middleman and just go straight to trying to find something that is directly capturing that, um, like how close to the events that you're detecting look like real ones versus just like fluctuation. Because even the noisy, um, like a lot of the noisy um, recordings still have events, but they just don't like I can kind of show examples of this of how the ones that detect more of like the greater than 4 standard deviations seem to have like a very stable baseline with big deviations, whereas the ones that are mainly less than 2 standard deviations or have a higher proportion of those as being detected as events tend to have like a more kind of noisy, less self-similar um, look to them, if that makes sense.
So are you assuming that, um, okay, I suppose this makes sense.
Yeah, there's definitely a lot of
How do you know
assumptions.
what so you've developed this algorithm on your own to merge events together, is that right?
Yeah, I mean, I'm not even sure if I would. It's like, I mean, it's basically just taking things that are within a certain uh, time of each other and merging them into one. So like here, where there's like a few that occur in a pretty short period of time, they get merged into this. That's where like the window that we'll define for our cert for our event on either side of this will be these. Um,
But
I think
then,
yeah.
may I think the idea is I think as I understand is to try to find the background where there's no activity in the cell and use that as a measure of the noise. And then uh, when there is activity, that's what you would qualify at activity signal. And so the intent is to measure how far away from the noise are the signals across the populations, across the experiments. And so
Mhm.
to plot that. I think that's what I understood.
Yes. Yeah.
so with the ones that are not merged, they're actually pretty decent. Like I can see the ones around 54, yeah, and they're kind of on the onset. Except for that very last one that you're showing here. I don't understand. That's not merged and it's not really on the onset, it's more on the peak. But the ones that are merged now, that changes because it kind of goes somewhere random in the middle. So I'm just saying that the event before merging, it seems like they're identifying the onset of this rise of the calcium transient. But after merging, we're losing that. Depending on what you want to do with them, that can be problematic.
I don't
Hmm.
think you were intended to have a high temporal precision, right? Just to identify two zones, right?
When there is
One
Yeah.
in
activity
the
versus not. So that was the main
That's
goal.
right. That's right.
Yeah.
That's all the goal is.
Yeah.
Hmm. And so it kind of is like if there are, say, like there's a lot of detected events right here, and then it turns out that those are actually not very, when we, see how um, far they are uh, from the baseline. If there a lot of them are less than 2 standard deviations from the baseline, then that'll actually sort of pull it more towards that cluster. So it kind of works in a way to like be like, here's like their activity is being detected here, but it doesn't seem to be beyond, you know, there's some, the event detection algorithm is seeing some kind of fluctuation here that it's trying to explain, but it's a not very strong, if that makes sense, or it's not super, um, so it kind of gets accounted for um,
Sarah
down
has
the
a
line.
question.
Yeah.
Yeah, yeah.
Sarah, can you
Oh,
speak?
Oh yeah, sure, yes. Because I'm a bit late and I have missed how you define your baseline. So I'm just a bit confused because some of these events around 40 seconds, I don't fully understand how they exceed the baseline measure. And so I was just wondering, maybe you could have a very conservative baseline and just say, what if you take, for instance, the lowest 10% of fluorescent values across the entire trace? I don't know whether they have a moving average or how you define basically this cutoff, because it's not clear to me visually what the baseline is that is exceeded for some of these events that you just discussed and say is a little bit noisy.
yeah. So the events are actually, they come actually before we calculate the baseline. So because the events are coming from the Allen Oasis style deconvolution algorithm. And then once we have those events already, then we calculate um, based on this kind of like slicing things into an event window and a non-event window, what the baseline is.
So then I'm confused. This is why would you go back once you have a deconvolved trace to a
Because a lot
non-convolved
of time,
trace?
yeah. Um, I think partially because a lot of times you get uh, multiple events in rapid succession, or it can sometimes the algorithm will try to infer events in like an area that actually is just kind of noisy, where there's a fluctuation happening. Um, and I think I know it's not like a, it's kind of a heuristic approach, but I do feel like if later on that there's kind of like some reason to like it does seem to kind of identify pretty high quality, at least to my eye, looking recordings and separate them from ones that are more noisy. I don't know. I mean, um, yeah. If
I'm
we,
just wondering whether,
Mhm.
maybe also coming from the other side, as I said, just looking thresholding the calcium trace itself might be able to kind of converge with the deconvolved event detection. So you could kind of find a common denominator that you think is basically, yeah, validated under both conditions, because probably both of them have different kind of issues, let's say like that.
Sorry, I had kind of trouble understanding you there.
So I'm wondering whether if you go the other way around and you basically threshold the events purely based on the calcium trace, you could maybe
Hmm.
compare them with, the event times and the number of events you detect based on that method and compare it with what you get from the deconvolved event information to then
Okay.
find like a common denominator that you think is kind of like the best compromise between the two approaches, because I mean, both of them have issues in a sense that they might not be perfect, but depending on the question, you might be able to just find a compromise that works for most things that you try to analyze.
Okay, yeah. Yeah, I can definitely um yeah, come up with a calculating the background first and then um going from there and comparing that to this measure to see if there's where they agree and where they disagree.
Why don't we let Davis show is how he derived those metrics, and then we can discuss over avenues to evaluate the background. I feel
Yeah.
like that would.
Okay. And so this just is kind of what I was talking about in terms of sometimes you get a worst case one where it's merged together, but the vast majority of them, of like these merges are within 0, to 1 second, so you don't end up with big slices that are being counted as event versus non-event. And then let's see here. Oh, I think that might be the same. And this is just giving more examples of some of the different windows. With these are normal, and then a merged one. And then some of the warning ones. So if it was a longer one, I flagged that to exclude it from the analysis later on. Because you can see it's kind of like something here where it's like it can sometimes just flag a lot of the deconvolution algorithms can flag like a lot of events in a back to back in a way that doesn't really seem super plausible. But when you have a part of the session like this, it seems much more reasonable. So you're kind of taking advantage of the fact that the algorithm finds these, that deconvolution algorithm finds these, because then they end up being less than 2 standard deviations, because they tend to be kind of smaller compared to these big ones. And you look at the proportion of how many of the ones that were detected were actually larger versus how many of them were smaller like that. Though there could be an underlying process that's causing this to transition to a different, baseline. So that's another thing to take into consideration, I guess. And I'll and this is kind of giving the distributions of how, like, an ROI, what percentage of its background were, I think, of the events were less than 2 standard deviations, 2 to 4, greater than 4. And then also kind of the exponential fit and Gaussian noise assumptions that we talked about in the last meeting.
And Davis, so the x-axis is what over what? What is the x-axis?
This is so for this one, it's like the fraction of events that were within this range of the baseline or the background. And then for this one, it's I think this one is R squared, and this one is
Sorry, I don't think I can see your mouse.
oh no. Okay.
Which... Oh, there I can see it now.
Okay, sorry.
Can I ask this question? So Jerome, you had written this in the email, right? So let me ask you this, please. So my understanding was that the background is when we don't have the events.
In
So
this
then what
definition that Davis, proposed, yes.
So then, but then Davis is saying this is the distribution of events in the background relative to not background. I guess I'm very confused here. So I thought
Me too.
background means the non-event portions of the trace.
Yeah, sorry. It's just I think I was doing this to define different measures of events, because in some of them I had the 95th percentile and things, whether it was above that. And this one, I think I kind of grouped them, I named them that because they were based off of comparing to that background definition I talked about. So sorry if that is confusing. But I think something that I might skip to something. Let me see if this that kind of, so where this kind of is going, that might make things a little more useful, is so basically after I calculated those events and which sort of bucket they fell into, of either being less than 2 standard deviations, 2 to 4, or greater than 4 standard deviations, I just use like a k-means algorithm to cluster them into different clusters, and then uh, made it so you can kind of like look at the individual traces here with the detected events. So like, for example, this one is like mainly greater than 4 standard deviations. Um, sorry, my own laptop's in the way here. And then you can kind of see that like the ones that are in this cluster tend to have like this relatively, like, to me at least, it seems like a very clear kind of transient behavior, um, and like versus ones that are more in this cluster, it seems like it's much more kind of noisy. And even though there's a lot of detected events, it seems like a lot of them might be kind of spurious. Um, and then also you can kind of see by plane, there's like definitely differences. So if this is the depth of the planes, um, then we can see that some of the ones, it's the depth axis. Yeah, you can see it's right there. Interestingly, I'm not sure if I did something. I tried to double check that this was correct, because I thought originally that the numbers after the layers were the depth order, but it does seem as though like at the 300 millimeter planes seem to have a higher concentration of these greater than 4 standard deviation style traces. And then the some of the, I think in this one it's VISI 6, which is like shallower, and then VIS 2, they seem to have less ROIs in general. So I'm not sure if I may have done something wrong here, but
No, I think the superficial plane is in dendrite, so you're gonna get very different kinds of events. So I think it
okay.
makes sense. You're like at
Okay.
the superficial layer, and so there, the kind of event are detected are very different kinds, right?
Hmm.
Of calcium event, whereas the others is somatic.
Okay, gotcha. So yeah, that's
Can you
definitely something to take into account. But yeah, if there's anything you want me to emphasize or look
So,
into
so
here.
you're saying in this k-mean clustering, the red dots here are the most isolated, cleaner kind of events for
Yeah.
soma, and then the blue are seems to be more often sort of hidden in the noise. Is that right?
Yeah, it seems to be more like they're not necessarily all bad recordings by any means, and it's actually most of them, most have a majority, or for most of the planes, yeah, they have the 2 to 4. So it's there's actually relatively few, less than, like in an absolute terms, less than 2 standard deviation events in any given trace, but it does correlate strongly, it seems, with them being more noisy. So most traces are a mix of the 2 to 4 standard deviations and the greater than 4 standard deviations. And so I decided to do a 3 cluster um, analysis here because that's kind of what we initially decided on. Um, and it seems to, it may make more sense to just do 2, but I kind of did. So mixed in ambiguous is kind of like if there's more of a mix of these 2 and 4, um, or 2 to 4 versus greater than 4 categories, and whereas um, the greater than, mainly greater than 4, there's a lot of those. Um, but yeah,
Sarah,
this one has a lot more.
makes
Um,
sense. Sarah, do you have a question?
Yep. I'm wondering, based on what you just said, Jerome, do we have access to the size of the ROIs? Because maybe it would be nice to plot this data just in relation to the size of the RI and then the signal to noise in a sense to see whether essentially the big RIs are the somatic signals which are more reliable, and to what extent you have smaller RIs which are potentially equally reliable occasionally, or are they consistently less reliable?
Yeah, we do have the soma. I think Davies.
Yeah, I don't think I have area as like a metric I can sort on implemented at the moment, but that's definitely something I think we can do. But yeah, right here I have, you can kind of see the as you go through the traces, it'll show you
I do.
which, yeah.
I do want to mention something important here is Goose mice are pan-neuronal mice, so you have a mixture of inhibitory and excitatory cells, and so they are in the layer one, they are, as the literature says, likely NDNF inhibitory neurons, and throughout the layers, you're going to have a mixture of excitatory and inhibitory neurons. The inhibitory
Mhm.
neurons are known to have very different kind of firing profile. And
Yeah.
so in your metric, that should show a little bit. I would expect the inhibitory neurons to have a less of a, clean baseline and higher, like they tend to fire more tonically.
Hmm.
So you should see that in your metric as well.
Okay. Gotcha. Because so do you think maybe we should use like more um sort of cell-specific um spike detection or
Or you
sort of spike inference?
you don't know which cell is which at this
Ah,
point.
okay.
So but it's just you're gonna see cells with a beautiful background and very isolated peaks, which are uh potentially pyramidal cells, and then some cells with like more tonic periods where it fires consistently for long periods of time, which are potentially more likely inhibitory neurons, but not always the case. But just saying they will distinct one another in that sense. And so your metric is going to be influenced by that a little bit.
Yeah, so it seems like I'm definitely capturing more of the somatic ones in that, and or
Yep.
kind of, yeah, I don't know if I'm
Which is
overhyping them, underhyping the other ones maybe more.
But remember, the goal of the figure, as we
Mhm.
laid it out, is to provide a first order description of the data. It's fine if it's a little confounded, but I think we just need to have a clear metric and then show according to that metric, this is what we see. And then in the manuscript, have a discussion about, why we see these things.
Okay.
Personally?
Can you show the depth borders?
The depth borders?
Yeah, I might be confused, but I see.
Yeah, I think
Yeah.
they're,
So, but I see these are the four now. So this is now
yeah.
1, 2, 3. And which one is the most superficial?
I think it's this one, the VISI6.
Yeah.
And then for VSP, it's 1.
But then why is
Or
it
no,
that we go from
2.
more neuropil to more soma and then back to neuropil? From superficial to deep.
Neuropil. The top layers is the layer one. So it's
So that
mostly
makes sense. But then
mostly dendrites and a few inhibitory cells.
Sure. But then why the deepest one is again more dendrites as well? Is that because the quality is now low and not because we have necessarily dendrites? So basically this is reflecting 2 points. If SNR is low, it's basically non-red means either dendrites or low quality.
That's right.
Okay.
You're getting deeper, you get more background.
Yeah.
And the signal is decreasing.
Okay. And what is GT for SD? What is that GT?
Sorry, that's just greater than.
Greater than 4 standard deviation relative to the background.
Yeah.
meaning that number of events during the activities for standard deviations more than the background.
Mm-hmm.
Number of events or magnitude of the event responses?
So it's if you take all of the events, you put them into those three categories, and then the proportion of how many of the events are any one category, or that's sort of, and then, yeah, does that make sense?
And then mostly 2 means equal to for SD, less or equal.
Yeah, so the other one is the sorry, zoom out a little bit. Yeah, so it's this is kind of the space that it's using to cluster
very
them.
nice. Yeah.
I feel like maybe it would be good to return to Sarah's point.
Mm-hmm.
I wonder if you could repeat this analysis with like a metrics that is less influenced by the event detection algorithm. Like I think she mentioned detecting the lower percentile of the trace and use that as a background.
Mm-hmm.
Is that what you had in mind, Sarah?
Yeah, because I think it's the most agnostic, right? And then you just choose how you want to bin over time. Essentially, you integrate over certain temporal resolution to define the magnitude of the events instead of the number of the events. And so I think it might be, yeah, I would say it's more traditional, and it might be better to deal with changes in baseline, like what you had as an example before. Was a nice example where suddenly you have lots and lots of events detected because essentially the baseline is just completely different and much higher. And I think you might be able to just then say there's one big event which has a big integral, which is, I don't know, three times bigger than what you see on average or something like that.
Mm-hmm.
And I would be genuinely curious whether your ability to detect these signals is influenced by the size of the ROI. Because if I'm thinking about exploring this data, clearly somatic signals are the first thing you would look at. But I would be also very curious to know if you're in a certain depth where there's less somas, if you can pick up local high quality dendritic signals. It's a bit like layer 1 would be a beautiful example, right? There's not many cells, but there's definitely lots of dendrites. Imaging wise, it's a place that you can image really nicely. And then I'm just curious whether these signals are more noisy because they're genuinely more noisy, or is because you're not dealing with somatic signals anymore.
Well, I might be the one that is introduced with confusion, but I think we use a soma restricted create line.
Then you have a size threshold. It's just that there's lots of little green things here. I think
That's right. That's right.
So I'm not really sure how this is threshold is.
There should be the event detection. Might have a size threshold, but I do see smaller things here. I agree.
Just from a quality control perspective, I think it would be good to know, at least to define what do you call an ROI, right? Or are you agnostic and take everything, but then see whether systematically the size that you consider has a role in terms of signal to noise, or whether it doesn't have a role.
Davis, did you use because I think we should have packaged a classifier output in the NWB.
I did not make use of that.
I think the classifier sort of says which ROI are somas and which are not.
Okay, gotcha.
But might help with this question from Sarah.
Mm-hmm.
Do you have more to show, or things do you want to discuss?
I guess I mean this is I think I just had some more traces that were just showing, things that were close to cluster boundaries versus things that were farther away, but that's about it in terms of what I had.
Okay. Do you mind if I bring back the figure?
Sure,
I
yeah.
want
Oh,
to,
actually, I
I
do.
want to,
I had a draft of the figure
oh,
using this.
you want to show that?
Yeah. I just have
Because
to. It's not in my
I,
browser, so I have to share it.
my interest at the moment is to just keep us on track to
Yeah.
to get this paper out, and then allow divergence later on for analysis.
Share my screen.
Sarah, yes.
It's just one comment. So in terms of the figure, what I really like about what David just showed is this interactive aspect of almost like a figure panel. That you could have a code like what he has, where we illustrate something in the figure panel, which is one illustration. But then if someone can actually instantly use the same code to go in and explore, essentially select the dots they're interested in and play around with it. I think that would be a super cool kind of example of how a figure panel can go, it can go beyond a static example.
I agree. I think it was really cool visualization you show.
Yeah,
Oh,
exactly.
thank you. Yeah, I mainly just made it to make it easier for debugging and things like that. Than having to.
We could have this part of the supplementary assets of the manuscript and refer to it in the main figure.
Gotcha. And then
Okay, can you explain your figure?
yeah, so to be totally forthright, I actually just put the a screenshot of Jerome's figure for uh, the slice that was related to Mesoscope into ChatGPT and said, "Could you generate a figure that matches this?" Um, and uh, so it kind of gives a little bit of it, and this is only, I think, for one session. Um, I actually don't know where this J figure came from or exactly what this part of F is, but I don't know. This was just kind of like a uh, something I tried to put together at the last minute. I don't if it. I feel like this is really kind of the more valuable maybe type of thing we might be looking for, or like the class distribution across areas and layers, and this if that's something like if we end up using this metric like a potential um, thing to help explain um, what it where what this looks like visually. Um,
As far as I know.
So the idea, this is then our measure of SNR. Is that the idea, Jerome?
It's one measure of SNR that we could use, yes.
So last week we talked about the quality control metrics, right? And it was like a couple of SNR measures that Davis presented.
Yep.
So I'm trying to link this meeting with the previous meeting and then
That's
link
right.
everything to the figure. So is the idea now to make the figure mostly about this measure, and that becomes the primary SNR measure for the QC metric, or are we trying now to compare what each of them says and use one of them or a combination of them? So
Yeah. Can you bring back the figures, Davies? Actually, I thought it was really helpful as a.
is this one or the interactive one?
Somehow it does not share your figure panels yet
Oh, okay. Let me try
anymore.
that.
The one you had a ChatGPT generate.
Yeah, let me see.
I think for Sunday, it's up to us to decide if we want one metrics or severals. The goal of this panel is to, I think, was like as I laid out the empty boxes. I think the goal was to just introduce people to the data assets, and so here the top row is showing the ROI and the extractions from them and how events are detected. So I think it's actually exactly what I had in mind. And then the second row is explaining a metrics that is derived on those traces. And so I think here the left is explaining the metrics and the right is just showing the distribution. And then the bottom one is showing the distribution of classes across areas and contexts. So I feel like in my mind, this is already helpful. And we could, if you think it would be helpful, add more than one metric in this panel. But the intent is to sort of, I think like probably here, the figure should highlight the depth because I think it's hidden. You already start to see that the certain planes have a higher number of low, right? And there's a pattern, like it seems like there's a low, it increases as you go deeper. I think that's what we're seeing, but it's not entirely clear for how it's laid out. But I think it's a great start actually, and then you could iterate it using your metrics and maybe try out to do this with other metrics like lower percentile, as Sarah said, or some of the metrics that you presented last week if you think they provide a complementary view.
So there is something I'm confused here. With zero and with the 0 and 4th plane. So those are the superficial ones, right?
I think there should be, I don't remember the numbers, but
See, I also thought it was supposed to be like that, but then, oh, actually, yeah, you're right. It might be superficial, but they have the dendrites. That's why
But then why do we
yeah,
have high?
I guess.
The high ones were the Soma ones.
That's a good.
yeah, you need to double check this figure
Yeah.
because I think this is like a 5 minutes work.
Yeah. Yeah.
Okay, and then the other thing that I want to say here about this metric as an SNR. So a layer or a neuron may have good SNR, but not be as much active.
That's
But
right.
this metric will call it low. It will give a low value to it, right? Because
I
this
think
is about the number of events, from what I understood.
I think it's the proportion of events that are within a certain
But
distance.
I asked this question, Davis, and I couldn't understand what you said. So is it the proportion of the number of events or the proportion of the event magnitudes, of the calcium transient magnitude during events in the two? Do you see my point?
I think I'm not the proportion of the events versus the event
Maybe if you just show
magnitudes.
the equation, it would be clear.
Okay.
I think Farzana, you're as I understand, Davis is measuring the background, like he's trying to extract the background standard deviation, and then define 3 classes based on the amplitude of the standard deviation of the background. And so a high here is a unit that have activity that is very far away from the background.
Okay,
Yeah.
but I was actually talking most about panel F, that the x-axis or the right, so panel F right side, he's showing the standard deviation.
Oh yeah, so like how so this is like for within one ROI. This is like I think how many standard deviations does a given event? So there's one, or what is it? Yeah, so if there's one event that's 40 standard deviations above baseline, and then
and when you
there's
say above baseline, you mean above the
this,
magnitude of calcium of fluorescence during background. That's what you mean.
exactly. Yeah, sorry.
Sarah?
Yes, I just wanted to add, I like the plot on the right panel in F a lot, but if I was to kind of look at this data and explore, it would be more helpful to me if this was basically showing the distribution of standard deviations across a population of RIs. So I would imagine that you could average this and say, on average, this one example RI has a standard deviation of, let's say, 15, you know, and so it's a very reliable signal basically. But then it would help me if you would show me that across the distribution, so get a feeling for the spread of like what's the fraction of RIs that have a very high signal to noise, what's the fraction that is kind of low, which is kind of a segue into your categorization in J, honestly. But it will give me a better intuition of the spread of the data, basically, without having categories in place first.
Mm-hmm. That makes sense. All the transcripts from all these meetings are recorded, right?
We record the entire thing.
Yeah, that's what I thought. Yeah. I'm just because I feel like there's a lot of I haven't been taking notes on all the suggestions. So that's good.
The meeting will be on YouTube.
Gotcha.
So do you think, I feel like you could iterate this figure here using the feedback we provided today?
Yeah.
And Sarah, are you able to, because I think you said you wanted to work on the EFIS side of this figure with your team, is that right? Or?
Yeah, basically we started just looking through the data books from what was it again? Yeah, exactly. We found a very nice one, and then it's a bit similar to this. There's lots and lots of metrics, and we were brainstorming which ones we would focus on, and we decided to focus on the SNR metric that is actually sped out as a part of a quality control metric, but we didn't fully understand what that metric was. So that's one of the things we actually wanted to ask, like, what exactly is the SNR for a single unit? Like, what's the measure that we're dealing with here? Because it wasn't fully clear to us.
Okay. Can you ask those discussion in the forum? And then would it make sense that we discuss this next week, or do you need more time?
Next week I won't be around, but I'll be back mid-August.
Mid-August? Okay. Right. So I'll try to reach out to see what we do next week for the schedule. But David, you're gonna iterate this figure in the interim?
Yeah.
Okay. Please. Any other question you have for us, or do you think you have a good feedback today?
Yes, I'll definitely go back over the transcript and go through all of the feedback. But yeah, I agree with, this was I think it'd be a good idea to have like another metric coming from the opposite direction of starting from defining a background and then defining events from that versus starting from an event and then defining the background.
Okay. Lucas, do you have a question?
Yeah, just a very quick question. Would you say it would make sense to apply essentially the same pipeline to the SLAP2 as well? Because we also get transmission events. I just wonder, it might be easier to digest the figure if it's related.
It might be. I think you could. I mean, I think what Davis presented applies, right, to SLAP2.
Yeah, because we were starting to look into events, transmission events, and yeah, so I think this might be nice approach.
Okay. I'll reach out to a few of you to figure out our schedule for the next few weeks for presentation wise. I appreciate that this is summertime and some of you are taking vacations. All right. Thank you so much, David, for presenting today. It was, I think, a very productive discussion.
Thank you. Thanks so much for the feedback, everyone.
Have a good day, everyone.
Thank you. Bye-bye.